it.
Hello again, and welcome to 3reate. I'm your host, Andrew Newman. 3reate features two types of episodes in a single feed, weekly and interviews. Each week, Nathan and I discuss current trends, our projects, and whatever else is on our minds. We'll also bring you fortnightly episodes where I interview folks across art, science, and technology, where we dive into their stories and get to the heart of how creativity drives them. Thanks for listening. Today we're joined by James Lloyd. Where does the podcast find you, James?
Hi, Andrew. So I'm based in Perth, Western Australia. I'm situated near the beautiful Swan River in Western Australia. And just to start off with, I'd like to acknowledge the traditional owners of the land, the Noongar people. This always was and always will be Aboriginal land.
Yeah, thanks. Are there swans then, if swan is in the name?
Yes, there are black swans that swim along the river, black swans, and I do have a lot of photos of them as I go for a walk. I work at the University of Western Australia, and it's situated on the Matilda Bay, the bank of the river, and so I love walking along there at lunchtime when I need to clear my head and taking photos of them. I also cycle along the Swan River into the CBD, the city centre, and so I often see them. You sometimes see dolphins as well. It's a really wonderful place and lots of wonderful things in the river.
Yeah, for the non-Australian or European folks in the audience, what does CBD stand for?
Central Business District.
I know that one threw me a lot when I got out of this. We just call it like downtown, even though this, like in a New York City, downtown is relative, but it's where the big buildings are and where all the business happens, right?
Exactly. And like to me, downtown always feels weird because town to me refers to a small place, smaller than a city. But we say the same thing in England. I'm going into town, but you really mean the city centre. So to me, the Australian way of saying CBD, Central Business District, that makes sense. And it's very clear, I'm going to the boring part where there's lots of people in suits during the weekdays.
For sure. It'd be good to talk through a little bit about, like, you're English, right? From the UK, England. How did you get to Australia? Or why are you in Australia?
I never expected to end up in Australia, but I'm so fortunate that I did. I was born and raised in rural seaside Yorkshire, north of England, and very much love Yorkshire, but being from a very small town and wanting to study science and be a scientist, just that area, that hometown was not a great environment for that, for various reasons. Interesting politics there. And so I got out, but I didn't travel far. I travelled just to one of the local universities in Yorkshire, did my undergraduate there in genetics, really found a passion for it there, really developed that further. And then I was very determined. I've got an idea of what I want to work on for my research career. And there was only one person working on that, so this in plants. And at this point, I'd got very interested in plant sciences as a way of understanding how do the ways you turn genes on and off, how does that evolve between animals and plants? And a lot of researchers were focused on animals because there's links to human health there. Right. But I was thinking, I want to know how it evolved, what the pathway used to look like billions of years ago. So by comparing animals and plants, you can get at that idea. And then obviously plants are important for agriculture and feeding us. So curing cancer is great, but if people are dying of starvation before you get to developing cancer, that's not very great. And we've got climate change really making our pathway towards food security more difficult to achieve. So I think there is great value in doing plant science research.
Well, and drugs come from plants too, right?
Oh, absolutely. So many of our medicines come from plants. And something that we are thinking about a lot is, can you make more medicines in plants and not have to go through other systems where you've got big fermenters with yeast or
like
human
the flu
cells
with egg, that kind of stuff?
Yeah. So can we find a more sustainable green way of producing other medicines that are typically produced with animal or microbe, fungi or bacteria type processes? So then I moved all the way from York to Leeds for my PhD, which is about 40 kilometers, about 26 miles. So I moved during my education, probably a total of less than 100 miles. And to me, that was a long way. And then suddenly I decided, I'm going to go to America for a postdoc, and went all the way from the UK to the west coast of America, the furthest part of America, except Hawaii, to work in California, spent 3 years postdocing there. And then I changed fields slightly towards synthetic biology.
Okay.
And that's when I moved to Western Australia as a postdoc. And then I've been working here for the past 7, 8 years. And I've transitioned from working as a postdoc to running my own research group. But I've kept working on this synthetic biology, which is, can we essentially rewrite the genome? Can we create new DNA sequences that are encoding something novel and change? So, for example, I studied the basic natural ways genes turn on and off during my PhD and my first postdoc, but I wanted to then see, can we create a new artificial way of turning genes on and off that's new to plants, that doesn't interfere with the natural way the plant turns its normal endogenous genes on and off, but we can control new genes we introduce in a very predictable manner. So that's what I've been working on for the past few years, and we've got some publications out on that and a lot of ideas on where to take that.
You said a lot. How does that relate to something like CRISPR, which a lot of people might have heard of, which is effectively a gene editing technology at a high level?
That's a really great question. Just to put it in context, so the normal way people think of CRISPR is you've got the natural genome of a plant. Let's say you take a boring plant like the model plant we use in the lab, Arabidopsis thaliana. It's the lab rat of the plant science world. It's not, nobody eats it. It's just a weed that you see growing in the cracks in the sidewalk.
Yep.
Now, that plant has about 25,000 protein coding genes, which that's the material for like allowing it to grow roots and leaves and flowers. And if you wanted to delete one of those genes or mutate it so it no longer functioned, you would use CRISPR-9 technologies to delete that gene. But basically, you're just making a small deletion inside one of the natural genes. There are ways you can use CRISPR technologies in really different ways. So you could use it to introduce new DNA, but we have various tools for doing that as well. And that's the normal route for CRISPR. You use it to make a small mutation, small deletion. Using the normal version, the default version of the CRISPR-Cas9 system, that's replicating random mutation that could occur in nature, but it's targeted in a way that you'd have to screen thousands of seeds, hundreds of thousands of seeds maybe, to find that change naturally, whereas you can target that specific change with the Cas9 technology.
It's kind of like a switch. You could turn things on and off. So like, a good example maybe could be like if a single gene controlled the color of the plant, you could change it from blue to green or red or whatever, right?
Yes. So when it comes to flower petal color, like if you had a nice pink petal in some nice ornamental plant, you could mutate that and maybe turn off the pink pigment being made, and it'll end up being a white petal.
Right.
The question to then change the color from pink to blue, let's say.
Yeah.
That's probably harder to do because you've then got to make a new chemical that is a blue color. A simple deletion of a gene might not be enough to do that. You may need to add new genes in, whole new enzymes that don't exist in that plant to then turn on. And you want it to turn on only in the petal because you don't want it turning on in the leaves. And A, it's a waste of metabolic resources, chemical energy being wasted. But two, you don't really want a lot of blue randomly accumulating in your green leaves. And so this is where actually the gene circuit technology I've been developing comes in. So it allows you to... So we've been able to introduce new genes into a plant for a long time, and often you'll just turn it on all the time. So it's got a part of the DNA, we call it a promoter, and it promotes the gene being turned on, promotes the DNA being turned into RNA that will then make a protein that will then allow the blue color to arise. So the gene circuit idea is we don't want an always on promoter switch in front of the gene, and we will instead, maybe we could use an always on promoter, but then we have a way of turning it off on demand.
Okay.
And then you would release that. So if you knew a particular natural part of the plant, a natural protein in the plant that allows it to only turn on in petals, we could take that, build a DNA sequence that would bind to that, put that into our what we call our gene circuit. So we would be able to allow that petal-specific factor, or it could be a heat shock factor. So it could be something that responds in high temperature. And we couple that with our circuit technology. That circuit technology will then bind to the region that we've had the promoter that should be turning it on, but maybe the default state is off. We then release it, but only in the petal, or only under the high temperature condition. We've done it with chemicals, we've done it with specific parts of the root, we've done it with temperature. So we've got those that they're published in my Nature Biotech paper from 2022, and we've got some other similar systems as well. So the system I built is similar to what you were describing in CRISPR, where you're able to make mutations with CRISPR, the normal default CRISPR system, and that's a permanent change in the DNA. Right. The way I do it, I use an older school technology called DNA recombinases. So the first recombinases were characterized decades ago, so predate CRISPR by a long way, and they're very good for targeting very specific parts of DNA. But CRISPR is good because it's reprogrammable. You can reprogram it to target almost any DNA sequence. This is very specific, so there's a natural target site of about 34 base pairs that it will target, which that usually doesn't occur at random in something like a human or a plant genome. And so you have to add that in, tailor it. So it's a bit more work to tailor it in. So this is why CRISPR was such a revolutionary tool, because that allows you to make a mutation anywhere. Whereas with this, you have to first add this site in and then cut something out. And that is a huge laborious process. But for us, we're okay with that laborious process because we have to add DNA into the plant anyway. So we're okay with adding in the sequences to target it. Both the traditional CRISPR and our system, it allows for a memory because it's a permanent change in the DNA, cutting of the DNA. It means that once you switch it something on or off, it stays like that. So this is why we call our circuits, some of the circuits I've been developing, a memory circuit. There is another set of circuits that I've been involved with. So this was led by a really talented researcher here at UWA called Adil Khan. So Adil took the CRISPR-Cas system, and he and other researchers have done this as well. They turned off the cutting part of that. So it binds DNA and exactly where you want it to bind DNA, but it doesn't cut the DNA. So we call it dCas9 for deactivated Cas9. So it just binds to the DNA. And the beauty of that is Cas9 is a big protein. It's just big, bulky. So you take it to a promoter, a specific promoter that normally turns on a gene. When it binds in the middle of that promoter, it often interferes with that being turned on. So he's now got a system where he's built what we call a NOR gate, which is on by default, but as soon as one of two factors comes in, it will turn it off. And so he can bring this dCas9 to the promoter and turn it off. So he's not using the cutting feature of CRISPR, but he's bringing it to the promoter to turn it off. And so he engineered specific plant promoters to be really good at binding this dCas9 and built up an array of circuits. So he built the NOR gate, showed that works, developed that further so it's more controllable, and then he started layering them. And as I'm sure you already know, Andrew, but the NOR gate is a universal gate. So it allows you to combine different logics. So just like in computers where most of the circuitry on your computer is either a NOR or a NAND gate that has a particular computer logic, his circuitry allows you to combine multiple NOR gates together to achieve different computer-like logic.
That's all. So CRISPR is like a one and done kind of thing, but the circuitry allows you to add different functionality, but then also trigger that functionality given certain conditions. You could add a gene into a plant where if it was really hot, it would do something a little bit different than if it wasn't really hot, then it would maybe bigger fruit or something like that, right?
That's exactly right. And that's our aim, to try to then couple this perception and response process to something of agricultural importance, something we're still working on. So we've got proof of principles in model plants that we can get the switching and get it in a precise way so it's only there under the heat conditions or only in one part of the plant, like a specific cell type of the root. But now to take it into an agricultural setting, that's the thing that's going to take a lot of development. And the reason we call it a gene circuit is because it does mirror the ways computer circuits will process things. So if you build an AND gate or an OR gate into electronics, the logic behaves the same way as what we're doing with our gene circuits. But we're not attaching electrodes or wires to our plants, but it's just the logic, the mathematics of a circuit that we're mimicking, but with our DNA that we're introducing.
Yeah, I think to try to bring, that's a really cool innovation, because I try to bring this down for folks who are not in the industry or haven't worked in the industry. I think Roundup Ready is probably a very big brand of Monsanto. And the idea with Roundup Ready was that you could put the gene into corn, so you could spray as much Roundup on the corn as you wanted. Everything would die except for the corn because it had a resistance. Vitamin D rice comes to mind, adding in vitamin D properties so that people who have low vitamin D intake can get the vitamin D from the rice. That's a one and done. I don't want to try to minimize how hard of a thing it was to do, but it's like a one and done thing. But you're saying the technology you're working on has a little bit more flexibility in terms of so you could theoretically make it Roundup resistant and some other chemical as well, so you could alternate maybe?
So, yeah, and so when it comes to something like the golden rice, where you have vitamin A overaccumulation.
That's vitamin A, thank you, yeah.
So you have a vitamin A overaccumulation in the rice, you get the beautiful golden color. And when you do that, again, it's always on. And they had to add multiple genes to get the plant to go from the chemical soup that it normally produces for what the plant wants to then produce vitamin A specifically. And some plants produce vitamin A naturally, but rice does not. And so being able to take the natural biochemistry and shift it, that is very beneficial. And it turned out that when they did that, it didn't have a big effect on the plant. There was not a big metabolic burden or growth inhibition of the plant. But if there was, then you may want to then turn it on only occasionally. So you may to allow for better growth. And so that's what the gene circuitry would allow. So again, like you said really well, was that the gene circuitry gives you flexibility and on where things are turned on, when things are turned off. Whereas the Roundup Ready corn and other Roundup Ready crops, again, they're always on. Right. Now, fortunately, that's okay because they don't have a metabolic burden. Right. Having this one gene turned on in those plants is not a big problem. But there are cases where having a single gene turned on all the time hurts the plant. Right. So a really good example, there's lots of these examples, is the immunity of the plant. So if you were to take a gene that fights a pathogen, like a virus or a bacteria,
fungal infection, beetle.
Exactly. And if you take the genes that naturally turn on in response to those pathogens attacking, that plant will be sick. It'll basically have an autoimmune disease. And speaking as somebody with an autoimmune disease, it's no fun. So you're continuously in this stress response state, and you end up with a little weedy plant that no farmer on Earth or anywhere else would want to grow. And so that it may be better at fighting the disease, but it's already sick from thinking it's fighting the disease. Okay. So if you could tailor that being turned on and off better, we hope to be able to have the best of both worlds, where you get the really good growth under normal conditions and you get improved growth under the pathogen stress conditions or any other conditions. It could be, as you said, like production of a useful chemical like the vitamin A. You could, if that becomes a problem, if you were to create a different chemical, a different medicine maybe, and that is a metabolic burden where it slows down the growth of the plant, tailoring how you turn that on is really important. So yeah, here at UWA, we've been working on a set of genetic tools to allow for this tailoring.
That's really fascinating. So I think for folks too who aren't maybe steeped in the field, there's always a trade-off in biology. If something gets bigger, something gets smaller, or it takes more energy to produce, so the plant is more susceptible to disease or less susceptible. And it's a very complicated thing. And I think science fiction has done a really great job of making it sound like I just kind of point to the genome and say, I want the plant to be green and turn it to red. And then I want the fruit to be twice as large. And then it also is going to be drought resistant. And why don't the scientists just make that happen? Because that's what we need. And then the reality is it's hard, right?
No, it absolutely is. And I love sci-fi. And this is why like working in the ARC Center of Excellence for Plants of Space is so much fun, because I love sci-fi. I love that dream of having a programmable plant where you can just, I can make it red, I can make the fruit bigger. That's the dream that we want to get to. But the reality is it is really challenging, because anytime you flip a switch in the plant from the natural 25 plus thousand genes, you're going to upset something. And I like the phrase context is king when talking about synthetic biology, because you can make a small subtle change in the DNA of a plant or anything, could be a bacteria, even bacteria which we think of as simple. You make a small change, but then, and you predict, it'll just change this bit of sugar metabolism. And then suddenly you see all sorts of weird and wacky ways in which it's changed the plant, because you don't realize how interconnected the natural wiring of how plants respond to the world is. Same with the bacteria, just the level of interconnectedness is wild. And so the gene circuits, we try to minimize that, because we think of it as something that we call orthogonal. And so basically, we're trying to keep it insulated away from the natural processes. So the 25,000 genes of a plant naturally interconnecting and doing their things. We want to minimize the interaction with that, insulate it away, and just add in one new feature. But even then, so many unexpected things can happen. We found that, you tweak this one thing, suddenly it doesn't work. And then you tweak this other thing and you're able to correct it. And we're continuously finding examples of this in our work, which it just shows that our ability to rationally design biology is difficult, because biology is, by its nature, irrational.
Right.
Evolution is the blind watchmaker, and it's done all the wiring without forethought of making it modular and easy to re-engineer. And so we're discovering all the wacky and unexpected interactions in biology through our attempts at engineering.
And that I think is a really good point to break down a little bit too, in that when you say 25,000 genes, that's also pretty small compared to something like a human, right? Or something more complex. And I think...
Well, actually, humans probably only have 20,000. So plants are arguably more complex.
But
it depends.
We've pissed off like half the people, and the other half the people have no idea why.
I love triggering people who go on about human complexity and stuff, so I'm more than happy to do that. But it also depends on how you define a gene, and this is a bit variable, because the easiest way to define a gene is a part of the DNA that will eventually make a protein. And that protein could be an enzyme important for making vitamin A or breaking down the food you eat. It's also the things that make the neurotransmitters in your brain, or the hormones that flow through your body. They do all of these things. They make the structure of your cell that allows your whole body to be created. You've got genes involved with growing a beard, or in my case, and I know that this is a podcast medium, so the audience can't see me, the lack of hair on my head, but the major amount of hair on my face making the beard, that's all encoded by different genes that are making these proteins that allow this, and then making the structures that allow for the hair to emerge. All of that is where the 20 to 25,000 genes are interconnected and building these. But those are the protein coding genes. You do have other parts of the genome that are doing things that are more structural, or they're making just purely RNA that is also doing something. And that world is a lot harder to study and to characterize. And so that's where some debate comes over how important some of that is, and whether humans are truly like the most impressive creation ever in the universe versus just one of many things that are very impressive.
And RNA for a long time was considered just like junk, right? It didn't seem like it really did anything by a lot of people. Recently, relative terms, right? The last probably, 30, 40 years.
I think so. The messenger RNA is what allows you to make proteins. So people, I think, saw that as just like a tool, an intermediary to make the protein. So the proteins were the interesting part. Yeah. The DNA was like the book that had the recipe for that, and the messenger RNA was just like the intermediate. But then there is examples where lots of other RNA is just made and never makes a protein. And that's where the debate really rages over, is this other RNA useful? And then there's all this other DNA that may or may not make RNA, and if it does make RNA, it makes very small amounts, and that RNA could be selfish. And so these are like the transposable elements of the genome. And if you bulk all of that along with some other repetitive parts of the genome, you get the junk DNA, the purely junk DNA. And then there's big debates over whether or not that's junk or all junk or mostly junk. And I have very strong opinions about that.
Yeah.
But there's a lot of discussion and debate on that topic, especially in the human realm.
And I think that adds to the complexity, right? Is there's all this stuff that it doesn't seem like it really does anything or might be left over, right? I've heard that quite a bit. But when you start adding things with cell circuits and that kind of stuff, I would suspect that you might start to see, especially down the line, all sorts of potential stuff that you couldn't account for. Because understand this stuff well enough, obviously research it, you're able to manipulate it in ways to do things, but you probably have no idea how some of this stuff works, right? And that's like the part of, I think, the heart of science in general, but also foundational science people don't quite understand. It's like, we can change the color of a flower. That's really well understood and really easily to reproduce. That's the other thing, is you might be able to change shit, but can you actually do it in a controlled way? It's like luck versus skill kind of idea.
And I think in this engineering of biology, you need both.
Right.
Like, you need skill to plan things out, but I will admit, like, I have benefited from luck. And I've suffered from bad luck, and I have benefited from good luck. Like, humans often, we focus on, especially I think somebody like me, my personality type, it's very easy to focus on, I've been so unlucky, bad things keep happening, and I reinforce the negativity pattern. But I've tried with this research to also actually be more balanced and recognize when I got lucky. Because my default assumption is, I'm a rational engineer, everything's just going to work first time, and when it doesn't, I feel bad, but when it does, I just assume that's the way it was intended. But like during the building of the memory circuits, like one of the weird stories we had was like the major, most commonly used recombinase in all of biotechnology, been used for decades. It didn't work in our context. It didn't work for us. And so I had, just by pure chance, used a different one to build the first prototype, just because that was easier to build the DNA for, just by pure chance. And that worked, so that gave us hope. So then I said, okay, I'll use the most commonly used one now I've got it built, and check that works. Turns out it didn't. So then we were scrambling, because we needed a second input to be able to build these circuits. And we didn't have a second input anymore. Like, what the obvious choice was no longer working, so I had to find another one. And this next one I was able to get tested worked okay, but the other three we tested after that were bad. 2 out of 6 worked in our hands, and it's taken us many years to figure out how to get the other, I think 3 out of those 4 that didn't work. We finally got them working to some extent, and we've got some more built up, and we've got a manuscript being written on that topic now. Cool. But that's taken years for us to figure out. So for that initial building testing, I've got to remember I got unlucky with this CRI, the most common recombinase, not working. But I got lucky that I picked a different one that happened to work in my system and was able to allow us to demonstrate that it worked. So we've got to remember, you're absolutely right, look, both positive and negative is really important in this, because it is so hard to predict changes in biology, changes in DNA, and how they're going to affect everything else.
And there's just certain rules that you can't change, right? Like, you can't just gene circuit me a tentacle arm, right? Probably, maybe.
Give me enough money and time, and okay, I think this is probably going to be me sounding a bit out there.
Yeah.
But my opinion is, if we're not breaking the laws of physics, we could achieve it. The question is, on what time scale?
Sure.
As long as we don't break the laws of physics, we just need to rewrite the local rules of an organism, of a particular cell. So giving a human a tentacle, I don't think is breaking the laws of physics. Now, given our knowledge of biology today, the answer is no, we can't do that. But we're not breaking the laws of physics, so given enough knowledge of biology and the luck aspect of it, we could get there. Now, ethically, no, that's never going to happen, and it shouldn't. We're not breaking the laws of physics, and this is where I like to look towards, where can we push the boundaries of this? Because right now, we're babies that are barely learning how to crawl, and what you're asking me to do is run a marathon. And I'm saying, like, a baby could grow up to run a marathon, but it's going to take a long time to get there and a lot of training to get there. And right now, we're barely crawling.
And maybe the first baby doesn't ever develop the ability to run the marathon, and that baby trains their baby, and then maybe that baby does it. And I want to break that down a little bit for folks too. So it's like, there's laws of biology, there's laws of physics. So you're not going to be able to make me walk through walls with gene editing.
Probably not, unless
Probably
unless the physicists tell me a secret that is new to them. No, that I can't even.
Quantum reassembly, it's easy.
Yeah, once the physicists work that out, then maybe I can program it into biology. But the physicists say no, we can't figure that out. So I'll trust them for now.
Yeah,
I don't trust physicists a lot, but I'll trust them for now.
And you're not going to be able to then like gene edit me like a brick arm. You're not going to be able to like have me grow rock.
Ooh. I have heard of people getting bacteria to make concrete.
Oh, interesting.
So, out there things, again, laws of physics, a brick hand would be pretty challenging.
Right. Like a granite hand, I guess. Maybe make it, because like concrete is rock, but it's a recipe of different materials. Whereas I guess what I'm trying to get to is the pattern of biology is organic, and you're not going to be able to probably have me add something inorganic to my body without some sort of technological intervention.
I think it's pretty challenging, but I do think of like organisms that do produce shells, and you know that. Well, again,
how do you define rock? Right? It'd be
How do you define rock? Because you can't get inorganic production from organic matter. Again, huge challenges. I'd say, I don't know if it's harder than the tentacle problem, actually, because tentacles would be a lot of like detailed organ patterning, you know, creating. But when it comes to the I think for me, if I'm thinking about this in a serious way, which I know you were suggesting as
I'm
more of a
definitely
jokey
not
sort of
thinking
thing.
about it in a serious way.
But if I was to think about it in a serious way, I think the difficulty in making a brick for a hand would be the shape of it and getting the shape to be worked out well. If it was a brick made out of concrete, like, again, people are getting bacteria to produce this sort of stuff. So I'm not going to promise you anything, but again, like, you could in theory do very wild things, but this is very long way down the track. Like you say, multiple generations of people raising up towards running a marathon, getting from the crawling phase, and we're still at that very first crawling phase.
Well,
so
we didn't even know DNA was a thing until relatively recently in terms of human history, and we didn't even know what it looked like, in quotes. You know, we didn't see it initially. We described it, right, and then we could see it until last 100 years or so.
Yeah, people were debating for years, like, what is the inheritable material? And I forget the exact year when people demonstrated it was DNA and not the protein, because the chromosomes of animals and plants, it's a mixture of proteins and DNA wrapped around. And people looked at the DNA and said, oh, this is so chemically simple. It's just 4 chemical variants repeated over and over. That's too simple to create the complexity of life. It must be the proteins, which have 20 different variants. They're all chemically vastly different with different chemical properties. That must be where the action is. And they were right that is where the action is. That's where the enzymes are. That's where the the proteins that make vitamin A and the blue colors and the red pigments, that's where that action comes from. But the DNA simple 4-letter similar chemical code repeating, that's where the complexity is encoded, and it's purely a information issue at that point, because you translate that information into that complex chemical language. But again, it was only it was within the last 100 years that we figured out that it was DNA that was the heritable material. A lot of people were suggesting it was structural. It was purely there to be a scaffold for the proteins to do their thing. And we now know, no, the proteins are often acting as the scaffold for the DNA and keeping it intact, because it is this information problem. And then we got the structural information of DNA, and so you had 3 back-to-back papers in Nature, the journal Nature in 1953. And Crick and Watson, as well as Franklin and others, did amazing work to understand the structural information, piece that puzzle together. And that then, once you understood, oh, that's what the structure of DNA looks like, now it becomes more understandable that it is an information problem. And that's what led them to be able to, and the whole field, to be able to understand how that information gets decoded.
And that's why when we say, is a tentacle arm possible, seems to be sure. Do we have any idea how to go about doing that? We have some ideas on how to start, but that could be a 1,000-year process for all we know, right? Because this thing that you're building inside of has really only been discovered / really understood by humans for maybe 100, 150 years.
And one of the challenges is because part of it is the predictability of if you make changes to the DNA, but every organism's different. Like, we could learn how all the DNA plays together in a mouse as a model organism, but that doesn't mean that it's true for humans. It doesn't mean it's true for plants, because the local rules have changed, because the laws of physics stay the same,
Yep.
but there's different routes to the same place. Two people can take different roads and both end up in the same city. One may take a bit longer than the other. And the same when it comes to, we're all using DNA, so the same starting point, but the rearrangements of that. Like, just from an evolutionary point of view, bats have wings, birds have wings, insects have wings. They all got to that endpoint of being able to fly with wings, but they got to it through very different ways. They use DNA as the starting material. They have wings as the end product, very different structures, different materials, feathers versus batty flesh versus insect, probably insect protein. You've got this big gap in the middle. How do you get there? They all choose different, very winding routes to get there. You would have to relearn that in each one of those systems. Each one of those is incredibly challenging. Like, understanding all the genes, all the timing processes is very difficult in each of those. And you've got to study that in birds, you've got to study that in bats, you've got to study that in insects. Then there's the diversity within insects, there's a diversity within birds and bats. And so you see how this then becomes an incredibly big problem, because there is no one universal law that says this is how you go from DNA to wings. Right. Instead, there's multiple winding paths, and the biggest differences are going to be between these ways in which they independently arose that. So that's where the daunting parts of it comes in, and where that pours cold water on my fire of, yes, I can give you a tentacle.
Yeah. Even if you could give me wings, I probably couldn't fly because I'd be too heavy, right? Because that's just one evolutionary path. But there's also hollow bones. There's also feather structures and skin structures and all this other kind of stuff that also has to be in the right path, taking the right turn. Otherwise, you have wings like an ostrich and can't fly.
Well, and wings, feathers first evolved probably for non-flight purposes, heat regulation, thermoregulation, or mating. And given human wings, yeah, I agree, that's easy. They won't be able to fly. That's a physics problem. I can't promise human flight with wings, but I can give you some really cool-looking wings. Again, give me enough funding and maybe multiple lifetimes worth of research. Given enough funding, I think biologists could do some really wacky and wonderful things. But ultimately, giving the human wings so that they can go out on a night out to the nightclub and show off their wonderful, colourful
like
wings.
Peacocking, literally, right? Yeah.
Literal peacocking. That is a lot less useful for humanity than solving problems of curing disease, feeding the world's population. I spend my days and nights thinking about how do we actually use this technology to help feed the world, more than I think about the cool things. Right. But because I'm a sci-fi nerd, I'm always happy to think about the wacky and wonderful applications.
And that's part of the creativity. If you're not thinking about this stuff, if you're not what-ifing and supposing, you're never going to make anything out of what is accepted. And I would, like what you were talking about in your different processes, you just happen to try a different process for whatever reason. It was available on that day, or you were lazy, or you were like, why not this? I'm more familiar with this, whatever. And it's like biology in a way too. It's like, how did we get to wings, 2 arms and shit? I heard you mention, I would take a little different angle here, but I heard you mention something about a space institute. Are you trying to grow plants in space?
Yes, actually we are. Yeah, I'm part of the Australian Research Council for Plants for Space. So this is a centre of excellence that has been established by the Australian Research Council. So it's bringing together researchers across Australia, and the centre has got funding from the Australian government to be able to build what the capacity we need to be able to grow plants in space to aid human exploration of the solar system. And we have partners, so we are partnering with NASA. We have great collaborators in NASA, and we do have collaborations with people, industry, and space agencies all over the world. And so this is a truly international project, and I'm really excited and privileged to be a part of it.
Awesome.
I started working on Plans for Space through working with my boss when I was postdocing here at UWA, Professor Ryan Lister. So he was part of the group that applied for funding, and then the centre officially started at the beginning of 2024. And so there are multiple researchers all over Australia in the centre, and so some of them, not just plant scientists like myself, we have chemical engineers, we have psychologists, we have lawyers. So the legal aspects of how do you get things in space and who owns it when it's in space, all the aspects around that. So we have people with that expertise. So in terms of multidisciplinary science that people often talk about, I would say that we are very multidisciplinary because we're not just plant biologists. There are a lot of plant biologists because plants is in the name, plants for space. Space plants, yep. So plants for space, it's in the name. So there are a lot of plant biologists, but we do have lawyers and psychologists, engineers in the centre who are bringing amazing capacity to what we're doing and giving what we're doing focus so that when we engineer the plants. And so there's a couple of ways in which the centre is tackling this. So part of it is, what is the physiological constraints of growing plants in space? So if you're growing things on the ISS, or if you were going to Mars and you're travelling to Mars, you're in microgravity. You're in, some people might call it zero G. There is gravity, but it's
there's the purposes. It's not like home.
It is not like home, absolutely. And so you get the weightlessness that you see astronauts in the ISS. And so when you grow plants in that, they use gravity to help control the direction of root growth and shoot growth. Light is also important.
Yeah, and I think maybe just, sorry to pause right there, I think in space there is no down or up. On Earth, we consider down being the ground and up being the sky with the sun. And so the roots go down to hold the plant, and then the leaves go up to try to get food to grow the plant. And that doesn't exist in space.
And if you're on Mars, there is some gravity. It's about 38% of Earth's level of gravity. But if you're on the way to Mars, if you're in constant free fall around the Earth, like in orbit, the satellite ISS, yeah, you don't know, there is no up. You cannot define what up is. And the molecular processes that plants use to define what is up does not work there. And we know this. People have grown plants on the ISS. We've monitored this. So I have a collaborator in the UK at the University of Leeds, Suruchi Roy Chowdhury. So she is an expert at understanding the molecular processes of how plants sense and respond to gravity, and understanding how that affects root structure, how branchy they are, what angle the branches and the main root look and grow at. And so that in terms of how you can grow in such a stressful environment is a big challenge. And then if you are in that ISS or deep space type situation where you've got zero microgravity, it's not just that one stress. You've also got higher levels of radiation because you're not being protected by a natural radiation shield that the Earth has. You'll also have higher levels of carbon dioxide. The levels of carbon dioxide on the ISS are about 10 times higher than here on Earth because it's so difficult to scrub that. Now, plants can play a role in that because they can obviously absorb the carbon dioxide in photosynthesis. So plants would be a benefit for this, but actually having that higher level of carbon dioxide, we don't fully understand how that affects how plants are responding to their environment. And so I met somebody who does work at NASA last year who's working on this problem and looking at how carbon dioxide levels affect how plants grow and interact with other nutrients. So I think of space as a multi-stress environment.
Sure. But
then if you're growing somewhere like the moon or Mars, there's a different level of stress because you've got some level of gravity, 17% of Earth's gravity on the moon, nearly 40% on Mars. So maybe it's not a problem on Mars. For the moon, that's still like less than 20%, less than 1/5 of the gravity of Earth. We don't really know what that would look like on the roots. So that's something I'm very interested in. But then you've still got a lot of other stresses there. And we're not going to likely use the dirt of the moon or Mars to grow plants because we can't call it soil. It doesn't have organic matter. So it's basically like trying to grow plants in shards of glass, is how I've heard some people compare it to. And so there's researchers in Florida who have actually got lunar regolith from the Apollo era missions that were returned to Earth. Tiny amounts of it because it's so precious. And they weren't able to bring a lot back. And they've taken small amounts of it and grown some plants in that amount. And those seedlings, the photos of them are shocking because the seedlings are so unhealthy. They're so unhappy. And so we're going to have really major problems being able to grow plants in that. So realistically, we're going to have to look on the moon and Mars towards some sort of hydroponics or aeroponic system rather than traditional like soil-based growing that we hear on Earth. We're going to have to be doing likely indoor vertical farming, which nicely ties into the type of farming that some people are doing here on Earth. So vertical farms, sometimes called urban farms, where you have a big building and you have lots of shelves with LED lights, ideally all solar powered, so it's all renewable, but you've got lots of LED lights that are being powered and you've got a controlled environment for your plant. So anything we learn about controlled environment growth for space will be very beneficial for controlled environment growth here on Earth. So that's going to be a really major thing that I think we've got these connections, these synergies.
Yeah, and I would think like density would be another one, being able to grow more plants in the same space. Like we have that problem on Earth now, right? The field only grows so much corn or so much wheat, and the more dense that we can make the plants, the way that change the root patterns to accommodate their ability to thrive with less sunlight, all those kind of things are directly applicable to here on Earth to either get more food of the things that are hard to grow, or as climate's changing or we see increasingly unstable weather patterns, that we have a better chance of being able to feed ourselves.
Absolutely. So I really do think that what we can discover for trying to grow plants in this harsh environment of space is going to benefit agriculture here on Earth. If we can grow, I've made jokes in the past where if you can grow plants on the red planet, the barren wasteland of Mars, you can grow it in the outback in Australia.
I could even grow it.
Oh, I don't know about that, Andrew. Sorry, I'm sure.
That's really awesome. And the thing that you're kind of talking about, so I guess I have two kind of thoughts. One, International Space Station has way more carbon dioxide. As global warming and the greenhouse effect happen, our carbon dioxide's going up. So that's an obvious parallel. If you can figure out how to grow better in that environment, it should theoretically directly apply to Earth. But going back to what we were talking about with the gene circuits, I also see a reality where maybe I'll just give you like my illustration of this possible future, in that you have a corn plant and you put in your circuits to say, on Earth, these are the strongest attributes we need to make the biggest, bestest ear of corn possible, with as dense of field as you could possibly do in really shitty sun and drought and all this other shit. And I want that same seed to be able to function on the International Space Station or some spaceship as we go to Mars. Oh, and by the way, I want those same seeds to also be able to be grown on the moon or on the planet once we get there. That's like the grand goal, or I guess maybe a possibility, right?
Yeah, no, that is definitely a vision, and I think we're going to get there in steps. I think we're likely to tailor. We'll have some seeds that would be great designer built for space, some designer built for Earth that are really good at withstanding climate change and the consequences of climate change. And for the ISS, we may have a different stock of seeds. Whether we have one stock of seeds that's perfect for all conditions, that is a vision. Whether or not that would be economically valuable, but from a scientific, from a curiosity-driven point of view, could we build something that is that universal? Yeah, that's definitely something that excites me.
Sounds like we're starting a biotech company.
That would be the dream. And I think in terms of what the future holds, I think the possibilities are sky high, literally, as part of Plants of Space. But at the same time, I do want to be very realistic. I don't want to oversell the technology, because I think there's been a lot of hype over the years.
Yep. AI is the same way in the tech industry. So I 100% get that.
And I think biotech in general, it's just a hard problem because the development time takes a long time. And we all saw what happens when Theranos, Elizabeth Holmes, all the dodgy stuff around lying within the biotech sector. Now, that's just outright, that's not hype, that's just outright lies. That was pure fraud. And that's just unacceptable. And we see that in other industries. But I think when people try it in biotech, that's going to fail, because the science, there's really hard limits to what you can achieve in the science. And you've got to be really skilled, and you've got to be very honest and open about the limitations and the timeframes for developing something in biotech are large. So the types of speculation I'm doing, yeah, I'm thinking this is going to be a really long timeline. When I say just because it's possible under the laws of physics, we might be able to build it, but I'm talking generations worth of research. I'm not trying to rush to that. Right now, I can turn on a glowing green, fluorescent green protein in a root and look at it down the microscope. And building up from there to being able to alter the plant in a really interesting way. That's what we're trying to do now. But again, baby steps. We're going from crawling to walking is what we're trying to do right now. But we're still a long way away from that really big vision. Just to, again, pour some cold water on the excitement of this.
And I appreciate that because it's really easy to hype this shit. Like I shit on AI people all the time because they make all these crazy promises that, yeah, sure, theoretically we will have machine intelligence at some point in the future that is way smarter than humans, that can self-replicate like the movie Terminator and maybe or maybe not kill all humans. Are we anywhere close to that? Not really. Realistically, no. Yeah. Like we're making advancements every day and things get more and more crazy and cool, but we're still so far away from that. And with the technology that you invented, it's like what, 2 years old, 3 years old, give or take? The early stages of even this, we can't even really get people to Mars, period, let alone grow shit there.
And getting to Mars, that's a huge challenge. And NASA's working hard at these problems as a larger international community of private and publicly funded people. And also, I just want to say, I've developed a system for gene circuits in plants. You've got Jen Brophy in the USA, who's developed some amazing stuff. Jennifer Nemhauser in America and her research group has been doing some amazing work in the field, as have many others. And here at UWA, we've got Ryan Lister, Adil Khan, and many other amazing collaborators that have been working on it. I don't want to take too much credit because, again, science is collaborative, it's a team effort. And not only that, but we stand, to paraphrase Isaac Newton, we stand on the shoulders of giants. And, other people have built amazing gene circuit technology in bacteria, in animal systems, and moving this over into plants was certainly not an easy project. It's something that took a lot of time and a lot of one step forward, two steps back kind of moments. But I'm really glad that we got there, and that I'm really happy that it has received some interest from the wider community in a way that a lot of my other research hasn't. I think it does capture the imagination of people in a way that a lot of science doesn't. And so the reprogramming of life, even if it is slow and laborious to do, I think it does capture the imagination. And it does feed into this other project of Plants for Space, because if we can reprogram the plants, I always sort of make the comment, if we did get to Mars and we want to establish a base there, ongoing human presence, well, there are no pharmacies on Mars. Right. So where are you going to get your drugs made from? And there's no oil on Mars. If there was oil reserves on Mars, then I think the US would already be there. And at the moment, we don't think there's any oil reserves on Mars. So where are we going to get our energy from? Where are we going to get our plastics from? So being able to build bioplastics that's from a green renewable source, because we don't have a lot of resources to exploit on Mars. Where do we get the drugs from? So we could make those in microbes, but then how are you feeding the microbes? You've got to then grow plants probably to capture the sunlight that you do get on Mars at lower intensity, but you do get sunlight. You capture the sunlight, you turn that into plant matter, then you feed that to the microbes. The one vision Plants for Space has is let's just build the drugs and the plastics and everything else directly into the plant and then extract it from the plant. And so that's where I've been involved with Plants for Space in particular, because bringing our technology to rewrite plant genomes and how they do things is really useful across the center for these very ambitious goals to, can we make the plant make us a useful drug that it doesn't naturally do? And then the benefit is, if we can get that to work for this hypothetical future on Mars, suddenly we built something that could be immediately useful here on Earth, because while we don't need to make the drugs that way here on Earth, it would be more environmentally sustainable, I think, to do that.
It could be a lot cheaper too, right? If you could plant your ibuprofen plant instead of having to synthesize the chemicals and bake them and however the hell they make ibuprofen, anti-inflammatory or anything else. And then a lot of drugs like insulin is probably a fantastic example, because that's basically grown today, right?
It is grown in genetically engineered microorganisms. And so we have looked at insulin and said, could we do it in plants? And instead of having a big bioreactor like a fermenter, almost if you imagine the beer industry in a big brewery type setup, that's what we're producing a lot of our drugs in right now. And so you've got to have temperature controls, you've got to have an energy source, sugars and proteins that you're feeding into.
And to your point, you're putting plants in there, just like beer is water and wheat and then some bacteria that eats it to produce the alcohol and the other flavors. That's a really cool and powerful idea, I think.
We think so. And I think there's some challenges. I think there's challenges to get plants to do what we want. Mm-hmm. Plants they don't have a literal mind. They don't have neurons, but they do have their own, to anthropomorphize the plants, they have their own desires and they don't want to necessarily be making drugs for humans. We have over 10,000 years domesticated plants to produce food for us in a way that is really productive, where modern day corn, maize, looks nothing like the ancestral corn, to the point where people had problems identifying the wild relative. And we've done that with other crops as well. And, and obviously that's an amazing thing. If you look at some of the original banana plants and how terrible they are with huge, horrible seeds, and then you compare it to the big seedless tomato plant. Sorry, I'm talking about bananas. So if you have the banana, you look at some of the wild banana plants with their big seeds and small fruit, and now you have these big banana fruits with no seeds. Suddenly that's a big change you've made through just years of inbreeding. But plants are going to be difficult to get to be making drugs very efficiently. And then there's actually a human-made problem, I think, of regulation. So the drug companies know how to work with the FDA to get a drug approved through their normal bioreactor fermenter type system. Whereas if you go to the FDA and say, I've made a drug in a plant, they'll be like, okay, what's the risks to humans? It's less risks. There's no human pathogens that can grow in these bioreactors because we're not using bioreactors. There's no human pathogens. It's just a plant. We eat plants. The FDA will say, approve it. Yep. Okay. And so it's not impossible. There's both biological reasons why I think it's a challenge, but then there's also regulatory reasons why I think it's a challenge. And so Plants for Space are not the first people to look at plants and say we can make medicines here more efficiently than in the traditional pharmaceutical approach. But we're definitely part of that movement and trying to push that forward. And we've set our sights on space as a way of sparking the community towards that and our research towards that. And we're prioritizing some drugs that are of particular interest. But any of the technologies we develop there, we will immediately, if we see value for it here on Earth, we will pivot it towards that.
It's like Plants for Space for use on Earth.
To help in space and to Earth. It's baked into our core mission. So we are very aware of how can we help life on Earth as well as help space exploration. And so we've got projects that will span all of that. And the more technology we develop, the new synthetic biology tools, the new switches for turning genes on and off we develop within the center, the better positioned we'll be to overcome the problems here on Earth, such as food insecurity, being exacerbated by the consequences of climate change.
And we'll never know what other stuff. So like the NASA space race, putting people in space was a really hard challenge. We learned all sorts of things that were easily applicable to life on Earth. Solar panels, for example, were invented to power shit in space. Can we turn photons into electrons? And spoiler alert, we can. Same with plants turning photons into fruit or vegetables or other compounds. So it's a really interesting problem. I think I always try to talk about NASA because a lot of the stuff that they develop, so Velcro is a really great example. Like, how do you hold people down in space? There's really no gravity, right? You're floating around. If you push off something, you're going to go flying. There's nothing to really slow you down. So they invented this sticky material that one side of it sticks to the other side of it, but it doesn't stick to everything else, right? I can put my clipboard down and my clipboard doesn't stick there. And people, I think, take that for granted, that nobody had any need to invent that on Earth, right? It probably would have never been invented. Who knows? It's hard to say, but probably not. And now Velcro's everywhere. I can't go through a day Velcro in my life at some point. And that's a really simple, easy to understand thing. But this is why the problem of space is very challenging. Trying to overcome that has direct application to our daily lives. And we then invent shit that we would never think about inventing because the constraints of space are so challenging. I love it.
Absolutely. The necessity is the mother of creation. So if you give a scientist or an engineer a really difficult challenge, as you've just elegantly stated, they will create stuff that is new to the world that would have benefits that people are not expecting. And NASA is full of these great examples, but I also want to bring it down to other examples. So here on Earth, a lot of the things we use often day to day actually started as purely to help people who are disabled. Yep. And so if you have sidewalks where it slopes down towards the road, that helps able-bodied people be able to walk across the street more easily without stepping or risk of tripping. But it also was invented to help disabled people. It primarily helps disabled people, but it helps everybody. I found out recently electric toothbrushes, and I've now my terrible teeth. I use an electric toothbrush as the recommendation of my dentist. And it turns out that they were first developed for people who have problems like actually with the motion.
Yeah, for sure.
And the electric toothbrushes are a great benefit and highly recommended for dental hygiene. But again, they were purely invented to help people who were disabled and needed that level of support, but they benefit all of society. And so by setting ourselves on a challenge that would help a niche small group of people, whether we're talking about people with disabilities or whether we're talking about astronauts, helping them in difficult situations then creates technologies that benefit everybody. So I do get very sad when I hear people, and I hear this from researchers here in Australia as well, who say any money spent on helping billionaires get to Mars is a waste of money that should be being spent to help solve climate change problems. I don't disagree that climate change is a huge threat, the biggest threat humanity is facing right now, and we need to mobilize resources towards that. But I don't see the Plants to Space Center or other efforts for space exploration as being counter towards fixing climate change. That money is not being wasted, not being diverted away from fixing climate change. It will create inventions that will benefit climate change. I have that strong belief based on evidence, based on seeing how inventions have come from the past. Thank you for bringing that up, because I think that's a really key point.
Yeah, and I think it's hard for folks to understand because we've spent some time explaining this really high level for folks who aren't familiar with it. And a lot of people are probably lost, and I apologize, but also go Google the words we're talking about and you'll find all sorts of information as to how to understand it. And that's like the curiosity part. But it seems really obvious. Oh, we need to be able to change plants to be less, or to be more drought resistant, or to be resistant to make weeding easier so that we get higher yields. And a lot of people in agriculture do this stuff every day. And that seems like a really easy idea, but the reality is to get to Roundup Ready or to get to vitamin A rice took 20, 30, 40, 50, maybe more years. And that's what I love about the Plants for Space initiative as well. As you see, it's psychiatrists, psychologists in there, and lawyers, and engineering people, and biologists. It's not just the plants. All of these other things that need to be invented or discovered, or problems from different fields have to come together. And then you get all sorts of synergies that you would never, ever see naturally occur because you're trying to hit this crazy problem. And then we get all sorts of other inventions to come out of it. And to your point, the gene circuitry problem, it existed in other fields of study, in different biology fields, and you put it to plants. And again, who knows, you have a system of doing it. CRISPR is another system of doing it that will probably inspire somebody else to try something different that will maybe create some new system that we've never even conceived of. And I think similar to entertainment, we talked a little bit about sci-fi, that awe-inspiring is a huge element of almost variability, and why I think science is so important. Art is really important as well because it helps push and illustrate these concepts. I was listening to you talk, like all I could think about was like the Star Trek replicator, right? They're reorganizing, for people who know what Star Trek is, they have a magic device that you can punch in some buttons or tell it what you want and it'll just do it. And the way that it works, in quotes, because it's a fiction thing, is that it rearranges things at a molecular level, right? And that sounds maybe insane to people, but the reality is everything around us is made up of 123 different things at a very small scale, the periodic table. Theoretically, you can rearrange that shit. When we rearrange it in some ways, you get, acids and water are not that's dissimilar. Or when you move some of those bonds, you create massive energy, which is like a nuclear reaction and a nuclear explosion. So it sounds really easy to say, oh, if hydrogen and hydrogen are the same thing, we could just change them the structure and now we have one thing or another. And then we put a bunch together and now we have something different. Way harder to do that. But we never get there without trying this stuff. And so the replicator technology, for hopefully most people are science nerds enough to know what I'm talking about. An alternative to inventing a technological thing is to what you were saying earlier was you could grow it. Like you could grow the plastic plant, and not that you're going to get a plastic bottle out of it. Maybe you would, but you're going to get the material that then you can more easily refine plastic from instead of having to dig it up from the ground or drill it up from the ground, because plastic is an organic compound. Oil is organic. You can make plastic out of other things. That's a whole nother conversation for a different day. But I hope that plants as replicators is this idea that I would love people to ponder on, because it theoretically, if you can grow an ear of corn, why can't you grow a cube of oil-like material to refine into something else?
Absolutely. And people are already taking corn plants and turning it into biofuels to power
ethanol.
cars.
Yeah.
And so we are able to move carbon atoms around a lot in plants, primarily carbon, not just carbon atoms. We're able to move them around and make all sorts of wonderful new things. So taking the product of photosynthesis, which is a simple sugar glucose, and then converting that into some sort of oil type product and then into bioplastics, that is definitely on the cards. And so I do love your analogy of a plant being a replicator. I love that idea. I have the same sort of dream. Can we get the plants to, if we reprogram the plants at the DNA level, how does that allow them to reprogram matter to create something new, like a useful drug, like insulin, or something like a plastic? Can we get that? And again, like for a while, we're going to have to be grinding up the plant and extracting that. And and then luckily we have technologies like 3D printers. So imagine we can produce the bioplastics in a plant, extract it, and then we can feed it into a 3D printer, and then you've got a nice enclosed local production system. And so we would envisage something like that on Mars, where that's an absolute necessity, but it could unlock potentials and democratize manufacturing here on Earth as well.
Well, in a really, I guess, easy in quotes, because it's not easy, but they use 3D printers right now all over in medical procedures, because everybody's bones are a little different. We're all human. We all have the same DNA. It's arranged differently. That's why some of us look differently than others. Bigger, taller, stronger, less strong, curly hair, no hair, right? If you can fabricate something, quote unquote, simply in like a 3D printer, you have personalized medicine. That's something that I've worked on a little bit in the past, and something I think people have probably heard about now in the zeitgeist. And it's very possible that in the very near future that when you need a knee replacement, you go in, they'll scan your knee, and they'll make you an identical knee for you. Andrew's new knee, Andrew's new left knee. And that won't work for James, but it'll work really well for Andrew. And that means there's less healing time. It'll last longer because it's not a universal knee joint, because everyone's knee is about the same. Range of motion may be a little bit different. The ability, again, we need to be able to replicate or print that stuff on Mars. That has direct 2-day applications on Earth. And to the point that we've been making as well, everybody who's making advancements on 3D printing on Earth also helps whatever space problems we might have. Being able to be in space and not have to bring with all this random shit that you may or may not need, but the raw materials, is a huge innovation.
And one thing that always amazes me when it comes to innovation in different fields is when you have these innovations in biotech and biomedical fields, they almost go under the radar, almost unnoticed. The improved tailoring of personalized medicine in surgery, that is amazing, but it's not like as obvious to people walking down the street. I'm only vaguely aware of some of these amazing changes and improvements. Whereas when there's a big technological revolution, like AI, like smartphones even. Yes. If I go back, I remember buying my first iPhone. I was 2009, second generation iPhone, relatively early adopter. I was very excited for that because I had the internet in my pocket now. Right. And there had been phones like Blackberries before that, but this was a new form factor, bringing new things together. And We saw now it's gone from a niche thing in 2009 to the default.
Yeah,
across
everyone
the
has
whole
the internet
world.
in their pocket now for the most part. Yeah, some people don't, but 99% of people probably do, especially if you live in Europe or America or Australia.
Exactly. And so social media, another example of an idea on the internet, to how you connect people, and there's different versions of that. We all use the umbrella term social media.
Yeah.
But the way it's connected people in ways that are both positive and negative. If you'd have asked me in 2006, when I was an undergraduate student creating my first Facebook account because it had just opened up in my college, my university, and back when it was still limited to a handful of universities globally, if you'd have asked me then, is this going to upset politics in a way that's going to cause all sorts of chaos globally over the next 19 years, I would have said, no, you're talking silly things.
Yep.
But the reality is very different.
For college students to spy on each other between classes, right? Yeah.
Exactly. That's what I thought it was. I didn't realize it was going to destabilize democracy. Innocent poor me in 2006. But I think in biotech, like, things move slowly.
Yep.
And when they are revolutionary, that's purely in the academic world. And then it takes a long time for that revolution in academia to become everyday in the world wider. But when it does reach that, it goes without a lot of fanfare, it goes without a lot of obvious noticing, but the improvement is still there. We're getting, some medicines are improving and getting cheaper through these background changes. And as you say, personalized medicine, surgery is getting better, diagnosis of certain diseases through genetics is getting better. When the human genome was first being sequenced over 20 years ago, the first draft was out 25 years ago, like, people had a lot of hype. There was the overhype of, oh, this will allow for personalized human medicine soon in a really major way that's going to change things overnight almost, and in a really profound way. Now, the thing is, it's taken 25 years for us to see some of these effects. Like, we have seen, there are newborn children who are being diagnosed with genetic diseases faster and better than ever before. But this is a minor thing that's happening in the background. It's very profound and helpful, or maybe even devastating, but new knowledge for the families involved with these things. But societally, it's a small change. It's important to many people in isolated situations. So the promise of the Human Genome Project has certainly not been fulfilled because the overhype was extreme. But the changes it's had in society have trickled down over the decades and has had a change in society and the way we think about things, the way we do things. But the overhype was toxic. And this is where synthetic biology, I think, is today. The overhype is extreme. The benefits are going to be real, but it's going to be a slow trickle of those benefits over decades. So here I am adding to the overhype by saying, oh yeah, humans grow tentacles. Headline, Lloyd says humans grow tentacles through synthetic biology. Again, I'm adding to this terrible overhype. But at the same time, I do think that we're going to have a lot of benefits trickling out over the next few decades, and that's where the benefit's going to be. It's not going to be tomorrow humans get tentacles, quote unquote James Lloyd. It is going to be a slow trickle of gradual improvements to the point where if humans did get tentacles, it's suddenly everybody's going to be, oh, that was obvious. That was the obvious next step of this technology in 200 years' time. Yep. Whereas today it's revolutionary, silly to propose such a thing. But in 200 years' time, it's like, that's the obvious next step because there's been this gradual improvement over time.
Yeah.
Because
100%. I don't think I don't think we could have had this conversation to a mass audience 10 years ago. Because I think maybe some people would have heard of DNA, like in a science class, or, oh yeah, I remember that. Genomics, probably no one really knew what the hell that meant or what a genome was. A lot of people still don't. If you don't, that's totally cool. But 23andMe, a lot of opinions on that, but they really popularized quote unquote DNA testing. And now I think DNA and what that kind of is much more well understood in the general population or in the zeitgeist than it would have been even 10 years ago. And to illustrate this point, a friend of mine called me. He had a kid a few months ago, but we talked maybe 6 or 9 months ago. And the doctor had talked to them about genetic testing. Like there was some reason to believe there may or may not be some genetic component was worth testing for, because precision medicine, genomic testing, there's a whole thing we could talk about there. He called me because I'm the only guy that he knows that has any remote experience in that at all, which very little I have, but I do know stuff. And we were talking about it a little bit, and I even remarked, I think, on the conversation, I was like 10 years ago, 5 years ago, maybe even 3 years ago, we would have never had this conversation, but it's already there now in that the kind of like the pathways of development. We understand that there's certain genetic conditions that we can get information about if we do genetic testing, and we understand even how to use the tool. We've been able to sequence a genome for a long time. It's been expensive, it gets cheaper, but now we're also understanding how to use that tool. So I always remark with a hammer, you can give anyone a hammer and they can pound stuff on it, but you can give a skilled craftsman a hammer and they'll be able to build your roof. But if they come inside and want to finish the trim around your doorway, they're going to maybe use a different tool. And that's like your circuitry technology is maybe that's more of a finishing hammer than a roofing hammer, than a sledgehammer, which is a different tool. And I think those are more obvious to people because I think the amount of information you need to know about just science in general is so vast. And then you have to understand the field. And to go get a PhD, we've had folks who have got PhDs who have talked about it. You have a tremendous amount of time and energy in a tiny little corner of a much bigger, broader, vaster thing. And to try to tie all this shit together is really hard and complicated, even for people who know what they're doing, who have spent the time, like you've spent the time, and you struggle, I'm sure, to find analogies.
And this is why I always enjoy speaking with you, Andrew, because you do have a nice broad range of interests and knowledge across different disciplines. And so somebody like me who is very hyper-focused in one small area and has a lot of deep knowledge in one area, it is always good to speak with people who have very different interests and have an eclectic range of interests to be able to put that in some wider context and learn interesting stories that are influencing other areas of technology and science and also just general society side of things. So I think that's a really important thing for us to keep in mind. And I do think about the ethics and the societal impact side of things as well. And as a scientist, like I'm not qualified to be a definitive person, right, authority on these things, but I am a person. I'm still a part of society. So
when you
I
have a
my
point
thoughts.
of view. And I think that's the thing a lot of people misunderstand about science is like, again, the tentacle arm thing is such a, I think, a brilliant example of this. Of sure, it's probably possible. I could go get another biologist who would probably say that's insane, or that'll take 1,000 years, or they'll have an opinion and a point of view. And because science is the way that it is, we mash all those points of view together in a field. We do things like papers and studies where we find evidence to support a point of view, a claim, and then that claim either gets built upon. CRISPR is a great example of something. They didn't just come up with that out of the ether. It's built on many hundreds of people's work over probably 30, 40 plus years. But it's really hard to demonstrate that without being in it. That's part of why I do the podcast is I'm generally curious about all this crazy shit, which is why I like doing it. But two, it's really hard to dive into this stuff without having that expertise, and why the focus of the podcast is art, science, and technology. Three things I'm also very interested in, but the technology field in general is massive. Science is massive. Art is massive. But you have to pick little pieces of that, latch onto it, try to understand it. Otherwise, you're never going to understand the whole thing. So if you didn't understand anything we were talking about today, I'll link to the papers in the show notes. And again, Google some of these terms. If you Google, like, what is a genome, or how does a human genome differ from a plant genome, you're going to find all sorts of information as to why that may or may not be different. Again, like we talked about, your point of view is that plants are a little bit more complicated than humans are. If I got a human geneticist, they probably... Okay, I don't want to put words in your mouth.
No. If I alluded to that, I guess it was a little bit of cheekiness.
Sure.
So I think what I'll say is, like, there is no correlation between genome size and complexity.
Yeah.
And this is like something that people have tried to wrestle with, this problem. Some people call it the C-value paradox. And this is jargon, and tangent on jargon. Jargon is considered bad because it limits communication. My view on jargon is a way of abstracting an idea. Like, you're taking something that might be a whole class at high school, a series of lectures at university, and then you're using a single word to summarize that whole package of information to other people who have had that same experience. Whereas obviously a lot of the listeners are not going to have that particular background, so that's why limiting jargon is going to be for the best, because they don't have that set of lectures from their education or reading the scientific literature to be able to understand what does that term encapsulate.
But to your point, it can also be very communicative, right? Because if we know the same jargon, we have the same experience, or we have similar knowledge and experience, we can go, oh, that's what you mean. Okay, great. And so I love that you're breaking down jargon, but then you're also breaking down the jargon you're using, because people who have either similar experience might not have the full understanding, or might not have any of the experience and have no idea like what we're talking about.
Yeah, absolutely. And I think, but going back to plants versus humans more complex, what I will say is the brain of animals, primates, humans in particular, is very complex. And it allows for things that are transformative in the world. So I'm not going to claim that is not actually like a very impressive thing. Right. But I remember I was on, I think it was Blue Sky, social media, where a scientist was talking about how do you define complexity? Because this is an endless sort of back and forth that happens in the scientific community. How do you define complexity? Some people might define it as, well, the number of connections within a brain. Then that definition of complexity makes no sense for a plant or a bacteria, which have no brain.
Right.
Another way of defining complexity might be the number of organ types or tissue types within the creature. So a single-celled organism, its complexity is very low because it just has one cell type, one tissue type, because it's just one single cell. Whereas if you're a plant, you've got leaves, you've got roots, you've got different tissues bark within the leaves and the roots. Bark, you've got all of the flowers, you've got all of that complexity. Humans, mice, have a lot of complexity. In terms of tissue types, mice and humans are probably the same level of complexity, but most people would say, I'm more complex than a mouse. Is that human-centricism, or is it an intrinsic thing? That's for both scientists and probably philosophers to get together and debate. So like this sort of discussion happens. I remember once recently on Blue Sky where somebody was asking this sort of question. I think I had said something somewhat jokingly, and I think they were unsure whether or not how serious I was. And I suggested, maybe we should define complexity as the number of chemical diversity an organism can create. This is sometimes called secondary metabolism, so the weird chemicals that are not part of core metabolism. And this is something where humans are really bad at it. We make a lot of very straightforward things that we need to survive that a lot of other organisms know how to make. Whereas these wonderful chemicals that often make up our antibiotics or anti-cancer drugs, what's really good at the production of those are some bacteria, some fungi, some plants. And they have an amazing array of things, and that's why we hunt for medicines in certain types of bacteria, certain fungi, and certain plants. And why, like, antimycin as an anti-malarial drug comes from plants. Aspirin eventually comes from plants. You've got penicillin coming from bacteria. Lots of other antibiotics, like I think lots of other antibiotics coming from bacteria in an arms warfare for there. So if you were to define chemical complexity as the core metric for overall complexity, then humans rank very low, and all these other single-cell organisms and plants suddenly rank really high. And to me, this is like how ludicrous it is to try to define complexity and say, humans are more complex than plants, or vice versa. Yeah. Because they're specialized in different ways. Yep. And I do think there's something interesting about complexity. Like, without multicellular life, you wouldn't have the transformations that you've got on Earth. Like, land plants have transformed Earth. We wouldn't have coal without land plants forming early trees and new compounds involved in that. We wouldn't have oil I think a lot of that comes from the algae in the seas, which, and the origin of photosynthesis that produces oxygen from carbon dioxide, that created one of the greatest mass extinction events on Earth. And that invention of photosynthesis using oxygen was catastrophically bad for life on Earth in terms of mass extinction. And obviously now we've got Earth populated by organisms that had evolved to overcome this. Now, obviously humans are a great example, great in the sense of like large and impressive, not good. they're great in terms of how we've changed the shape of the earth. We've built buildings, we've built great architecture, we have changed the atmosphere. We've done these great things that have both positive and negatives. There's positivity for, I like living in my apartment with a roof over it so I'm not exposed to the elements all the time. But also, I'm very sad that we're causing another mass extinction event through these byproducts. And the human intelligence and our ability to manipulate things is impressive, like on a global scale. So if you were to measure complexity or impact through the ability for an organism to change the environment at a global scale, then plants are up there, but then so are humans. And there's lots of different ways to think about that complexity, which this is probably just a random rant that is weird to have come up in otherwise, but it is something I think about a lot in terms of...
I think you make some really good points, and I think, too, it's also a matter of perspective that you look at. It's really easy. I think, like, building a building that's more than a couple stories is a very hard and complex thing to do. And we just take it for granted that 50-story buildings exist. And the people who have that perspective, the engineering know-how, the materials, like you can't build a 50-story building out of hay bales, right? It just physically won't work. And the same comes through with plants, is that a lot of the perception and the perspective that we have is limited by what we can see, touch, taste, feel, hear, our senses. But there's a whole world, like the subatomic world I'd like to talk about all the time. There's a whole bunch of shit going on we didn't even know existed until a few years ago. And we continuously discover more. And then we use, and I think what you're hitting on too is another problem of like interdisciplinary jargon, or even just like interdisciplinary communication. What I think of complexity in terms of systems is very different than probably what you do. And it's also, I see this all the time in business, that we're talking about multiple different fields, and we're talking to people who have some exposure, no exposure, maybe expert exposure. In business, this shit happens all the time, where you're in a room with 5 different people from 5 different teams doing 5 different things. And I've been in so many meetings where people are using the words that are interchangeable by everyone, but they're not actually communicating, because they're communicating their idea in their way using the same word as somebody else would use to communicate a very different idea in the same way. But because humans are also really lazy, they hear the word and they go, oh, I internalize the meaning to mean what I mean. So there are people who get too literal, but when you're talking about research, when you're talking about science, when you're talking about innovation, when you're talking about invention, you have to spend an inordinate amount of time even defining the shit that you're using to describe. And I think a lot of people just take that for granted. And that's part of a PhD. What does the vocabulary mean? What does the vocabulary mean in a specific context? To go back to that jargon discussion, if you're a biologist and you're talking about the nucleus, you're talking about the part of an animal or plant cell that contains the DNA. If you're a chemist talking about the nucleus, you're talking about the nucleus of an atom where the neutrons and protons are held. So in my building, where we are the School of Molecular Sciences, so we have both chemists and biologists and biochemists walking down the corridors, you don't know necessarily which type of nucleus somebody is talking about. But then, so that's a cross-disciplinary sort of issue. But then even within a discipline, I don't know how many of your listeners will be aware of the word epigenetics, and how much that carries. And that word has so many definitions, even within the field of biology. And I published on epigenetics or epigenomics related stuff. I'm fascinated by it. I also think there's a lot of overhype in this sort of subfield of biology that is also toxic, and there's some really fascinating biology going on there. But I forget that there's a researcher who has published online like a wonderful guide, a table and it lists like 5 different competing definitions of epigenetics, and it lists like who coined that term, when, and what that term means. And it's amazing to see like that diversity of what people mean when they use that same term. So I actually try to avoid using the word epigenetics as much as possible, because even within a field of biology, you can say this is an epigenetic phenomenon, and it might have, in a room of 5 people, it may have 5 different meanings. Yeah. And so I think language is supposed to allow you to communicate what's in your head to somebody else's head. Yeah. But a phrase like epigenetics, I find, is no longer fit for purpose without additional context to narrow down that meaning, even within a field. And that's not to say either that any of those, again, perspectives of philosophical or scientific discovery is right or wrong. It's also very perspective driven. And the cool thing with science is that over time, given enough people, the better perspective or the most common perspective or the most, I hate to use the word true in the same kind of word soup that we're doing, but come forward until something else comes along and then unseats that as the predominant perspective. And I think for most people in most of the world, a car is a car, right? My job as an accountant is an accountant until they're not. So it is very applicable as well to a lot of what people do, but we're talking about levels of perspective and specificity that are so much more detailed. It's very difficult to try to get that across if you're not familiar with it. But I also get really frustrated because it's so universal. There's multiple different types of accounting, just like there's multiple different types of biology. And then within, say, forensic accounting, you're going to have all sorts of subspecialties. And for people who have no idea what an accountant does or how accounting works, they're probably like, what the hell are you talking about? People who are accountants are probably like, this guy doesn't know what he's talking about. That's what I love about the podcast too, is I get to play the dunce sometimes. And sometimes I don't genuinely know, or sometimes I'm trying to make a point. But the system of science makes it such that all of this stuff becomes clear at some point. And I talk a lot about how the theory du jour or the latest paper in a field often gets picked up by the media. And then they talk about salt is good, salt is bad. Too much salt is this measurement. Too little salt is this measurement. And there's generalizations there for health and not. But the reason isn't necessarily that science is wrong or that the person before was wrong. You have to get into the details. We talked a little bit with Dimitri in the past about how to read a scientific paper, and the level of nuance in that paper is very important because you're probably making a very nuanced opinion. And I think our society in general has just lost nuance. But that's all science is, right? In this instance, your gene technology works super well to create a circuit. Doesn't work in people, probably. We're not going to test it because it's unethical, but doesn't work in rats. Or if it does work in rats, you'll have to do it in a different way with a different level of nuance that you probably have no idea about because you're not a rat or a mouse person, but someone who is there, you could cross-collaborate and maybe figure it out, maybe not.
Context is king.
Yeah.
It comes back to that quote, context is king. And so you can get something to work. Like the biggest challenge I'm going to face in the near future and tackling now is how do I move my gene circuits from a model plant that is the weed we talked about earlier into other plants like crops. And then, but not all crops are the same. There's more diversity between crops than between my pet plant and some of the other crops that are closely related to it. And so how big a jump in over evolutionary time are you taking is going to have profound effects on that. And then moving it from one, like, honestly, like you're talking about taking it into rats, but I've taken influence from sort of like mouse research into building these. Even with taking that influence, it's still taken years of development to get it to behave in plants the way we wanted it to. And hopefully some of the innovations we've created could translate back into animal systems like mouse or rat. But that's going to take those domain experts who understand the context of those organisms to do. But yeah, you're absolutely right there.
What's something that I should have asked you that I didn't?
Oh, good question. Because at the moment I'm starting up my own research group and going through a lot of fun but also stressful times on that. So that's been the sort of one of the biggest challenges of this year for me. It's a really rewarding time, but at the same time it's taken a lot of distraction away from the pure science and into sort of managing things. So if you wanted to ask about that's the first thing that comes to my mind.
Interesting. Yeah. So how do you balance then because you still have to do science. So you're starting up a research lab, and that means that you are no longer doing the research, and you'll presumably be hiring people and collaborating with people at a different capacity than an individual. But you still also have to do the work, or you have to do some work. So what does that look like for people who don't know what a research lab even is? Are you all in there with your white coats doing different colored test tubes of liquid and boiling shit? How does that look?
And like my plan for the next few years is probably to try and stay in the lab as much as possible. So I do want to be doing as much of the science in the lab as possible. But yeah, I have just hired my first two postdocs, so researchers who have finished their PhDs and now doing new science. So we have some funding for them to do some new synthetic biology work in plants. So hiring people for that's been super exciting. So I guess I like the anatomy of what it's like, of what is a lab, and so you'll have like an academic who runs a research group, which is the role I'm sort of evolving into.
Kind of like a CEO, now, right? Like,
I guess it's kind of like a CEO. Like it's
a
more
CEO
like
who also empties the trash and does the work.
It's definitely, I think it's better. I think of running a research group as like running a small business. It's like you're in charge of a cafe or a bakery. And so you do have to do those, take out the bins, especially before I hired my first people. Yeah, it was my job to take out the bins. So I had students and then there was me and there was nobody else. So it was like, I'm the one who knows the waste protocol off by heart because I've done it for years. I'm going to take out the bins. I'm going to do this. The amount of de-dusting I've done as part of this. Yeah, so you've got to get your hands dirty. And so it is either more comparable to you running a small business where you're dealing with the stock issues and supply issues. And
someone calls in sick, but you got to give that paper presentation.
Yeah, somebody has to do it. If something's time sensitive, then somebody's got to do it, even if somebody else is sick. And so you've got to step into these roles. You've got to do a lot of the planning. So, like, I do have, unlike a small business or many small businesses, I do have a wider ecosystem that can help. So, like, I do have a head of school or head of department that I report to. And there is the university infrastructure that is there to support it. But within that system, it does feel like I'm a small business owner trying to start up. And so in terms of, you've got the person who runs the group and they've got some sort of office and some sort of salary, but then you've got to have the physical space. And so I've got a very small amount of physical space in the lab that my postdocs are working at, and I'm working at. And so this is great to have. And so this is part of a wider lab area at the university that's shared communally. Like, we have a small area that we call our own, but then a lot of shared equipment, shared resources that we can all use and share. And we just have to act like mature adults in terms of how we treat those shared resources. And that's part of the privilege of being at a university where you have these shared resources across research groups, across departments, and means that you don't have to spend $20,000 on this particular machine because other people have chipped in together. Whereas I will buy some machines that are purely for my research that I may be less happy to share, or I might need to double check about sharing because I might need it at full capacity. My default would be, yeah, I want to share as long as you're going to treat it with respect and I don't need it at that particular time. Go ahead, please do use it. And that's how a lot of researchers do work. And I think this benefits us all in that shared sort of space. So that's where it differs, I think, from running your own business. But yeah, I've had to learn the HR process at the university to hire the advertised positions, hire people. I've had to navigate the purchasing systems. How do I create the financial accounts at the university and keep track of the spending on different research grants so I'm not overspending? So it's been a lot of learning in that regard, and I think I'm getting a lot better at it. And also juggling all of that with taking on some new teaching responsibilities and preparing my first lectures to deliver at the university in the genetics major. All just really exciting stuff. Like I've dreamt of this type of position for a long time. I've worked towards it since before we met years ago. I've been working towards this, and I guess there was always that concern, like maybe the dream won't live up to the expectations. But I'm actually finding I'm really enjoying it. I'm really thoroughly enjoying it. Now, there are days which they'd make me, if I had any hair left on the top of my head, I'd be ripping it out in frustration because there's certain paperwork that is frustrating to do. There's certain concerns that you have, or you're concerned with, what will happen if somebody turned down a job offer that we were going to make them, because I was really hoping that person who's perfect for the role will join us. So you've got all of these sort of concerns and worries, but they're outweighed by how amazing the opportunity is to be able to work with amazing people, hire amazing people, train great students, and have some independence over the science that you're doing and the way you're conducting it, in a way that I've had a lot of freedom and privilege to be able to conduct research and really exciting research. But now being able to have some independence, rekindle some of my old research. So I've just submitted a paper that relates to work back from my PhD days, like it's continuation, evolving of that research. And I'm hoping to continue with that as well. So not only work on this synthetic biology side of things and expand that further, but also revisit some of my older research that's been on pause, like it's been put on ice. Now I can take it back off ice, defrost it, and start going ahead with that and studying how the natural genome evolves and natural genes turn on and off and get into that again. Having the independence to go in all of these directions all at once is great, but again, maybe I'm a bit distracted because I'm trying to go in too many directions at once and I should probably refocus. But I've enjoyed it so much.
And that sounds a lot like even a typical startup problem. So like you have the university infrastructure you can plug into, or maybe it gets in the way sometimes too, because you don't have total freedom to go do whatever you want because you have to work within that system. But I don't think that's any different than in an art world where you're at a ceramic studio and they have the wheels that you use, or they have the clay that you can use, or you can bring your own clay, but then you have to go through another process. Or when it comes to fire the clay, you have to either hit a certain set of criteria or size requirements, or you have to rent the kiln for your own use. In a startup business environment, very technology, right? You're renting a computer system in a data center, right? You're probably not buying a data center out the gate for your startup. Maybe you are, if it depends on your startup. But so that's why I love talking through these different fields, because it's very different, but at the same time, a lot of the challenges, you're effectively a startup business owner who, instead of having the luxury to get a bunch of money from someone and just sit on stuff, you also have to operationalize it today. And there's a lot of laws and requirements in the grants in a business setting. If someone gives me $1 million to start a company, like I have to work with them and build something valuable. But in a granting world, you have to go even further than that and prove that what you did was valuable and that it was actually as valuable as they think it should be so that you can get funded again. It's not good enough just to get a few customers on your website or your app, right? So the challenges are similar, but also really different. So I love that, the comparison. And I think you hit on something that a lot of people don't really explicitly say, is that there are good days and bad days like everything else, but you have to really love what you like to do or you'll quit. And you would have quit a long time ago. Like you went to, how long did you go to school for? 8 years. And then you did postdoc, which is like on the job practice, even though you're an expert. And then you did a bunch of other work. And so you're 15, 20 years into your journey?
Because that's the thing.
Startups are hard. Startups take 10 or 15 years to materialize. It's a crazy career.
And like in terms of salary, like I could have been making a lot more money outside of academia, but I feel so passionately about this work and I'm driven because I love both the education and the research sides of working at universities. And I'm doing more of the teaching now, although still minority. I'm still primarily research focused, but I love the fact that I'm around students to train. That's the intersection between the teaching and the research side of it, is training up research students. I love being in that environment where you're passing knowledge on to new people, training people up in the art of research and your discipline specific knowledge. And if I didn't love that, then I would have gone off somewhere else and earn more money and be able to pay the mortgage off quicker and all of those things. Yeah. Like I'm so passionate about this that I'm willing to do it. And even a dream job is going to have good days and bad days. And that reality, like the idea of if you do your hobby as your main time job, you never work a day in your life. Exactly. Like I've got very mixed feelings about that saying, because there's still hard work involved in making your dream, your hobby, like your full-time business. There's still going to be, I don't want to fill out this particular piece of paperwork that I've got to send off to somebody. It's really boring. It's really written by lawyers, and I don't know how to read and write legalese. That's not what my science training was about, but it's part of the ecosystem and the legal requirements involved, so I've got to do it. But it's not my favorite part of the job. It's if you run a bakery, taking out the bins at the end of the day, so your bakery isn't filled with moldy leftover bread. That's part of it, but I don't think anybody dreams of taking out the bins when they're dreaming of becoming a baker, but it's part of the maintenance.
Yeah. And also, typically when you talk about it, you don't talk about it. Everybody always likes to talk about the positive result or the fun. And if you truly are doing what you love and what you're good at, it is fun. But you also need to acknowledge that reality that it is. There are days like I don't like to get up and get out of bed and do the work, or sometimes I don't want to do an interview on the podcast, but I generally love it. And I find also once I do it, or when I'm doing it, it's I guess with kids and parents, right? Sometimes you hate your kid, that you just can't help it at some point, but to love every stupid thing they do. So hard to talk through that because there's so many people who either don't have the opportunity or don't have luck. Like we talked a little bit about luck in there's so much of life is luck or being born in the right place at the right time. And if you were born 200 years ago, nothing would have happened. You would probably be a farmer, whatever the hell people did 200 years ago.
Even 100 years ago, that's what my family was doing. Like my mother's family, there's a lot of farming going on in that side of the family. And if I hadn't have died during childbirth, which is really the reality of 200 years ago, I would have been in farming or fishing or something like that. I wouldn't have had these opportunities. I'm dyslexic. Even if I'd have had some level of educational opportunities, I'd have just been considered a dunce from the beginning and not given further opportunities.
You probably wouldn't have had the ability to learn how to read.
Exactly. And being the look of where and when I was born, I'm incredibly privileged, but how privileged I am is relative because I'm dyslexic. I was born into a relatively poor family that has had no higher education in an economically depressed part of the country. But I was also born in one of the very rich countries of the world where there are great universities, and there was a pathway for me to be able to get into that education system. So are you privileged or not privileged is not a binary. And I'm definitely on the privileged side of things. I'm a white dude from a wealthy country, so already I've got a pretty good starting position just through pure luck. And so I do have a strong view of, no, we do need to make sure that opportunities to be able to get education, do science, should be as open as possible. And we need to democratize the technology as much as possible so more people have access to something. And there's only so much control within the wider system that anybody can have. But I love any efforts that can be made to make education, knowledge more out there. And I'm of the philosophical opinion that knowledge should be shared as widely as possible, because that is how society develops, how we develop knowledge, how we develop smart people who will create better things for the future.
And I think that's probably why we get along so well, because I totally agree with you. And the one thing I'm hearing lately in America, which is starting to really scare me, is that the systems like financial aid are under attack because they're expensive. And frankly, student aid in the United States is actually the second biggest revenue generator for the United States after taxes. So taxes are the number one way the government makes money. The number two way the government makes money is student loan interest payments. Just facts. But there's a lot of very wealthy people, ironically, who don't think that's a good system and think that might inflate the cost of education. There's some truth to that. Dive into that at maybe a different time. But the point of that is anyone in this country can take advantage of that system. And so many other countries don't have that, or they don't have the amount of money, and you have to compete to get a seat at that. And in America, there's some qualifications for financial aid, but if you're a citizen and don't have a ton of money, you probably get financial aid. That's so rare. And because of all of these other things or these other perspectives, people are trying to take that away. And I guess the really large point is that all of these systems of equalization are inefficient and cost money or cost time or cost energy. They allow everybody to at least get started. You might not be the best scientist ever, but you can get some money to go to school to maybe become the best scientist ever.
And also bring new perspectives. I think having people from diverse backgrounds, you're bringing a new perspective. Like that, I've heard, this is not my area of science, but I've heard people talk about how thinking in different languages and being raised in different languages also shapes the way you process things and process the world. And so having people from diverse backgrounds, diverse languages, and all of these things will hopefully mean that when we have constructive debate within science, because you talked a bit about this earlier, when one study says this, another study says that, this is how science evolves, through constructive debate, not destructive like bullying or criticism, but through, I think this based on the evidence, you think this based on the evidence, now we have to work out, test new things, get more evidence until one idea works out, or we're able to synthesize them together into a joint
somebody with
unifying.
a different perspective comes along and has a different, right? And that,
absolutely,
that 100%. And so I've worked in science, I've worked with scientists, and I just think that because a lot of people, at least, and I can talk about the United States, I can talk a lot about different countries too, but the people in the United States lack the perspective of what it's like in a lot of other countries where you don't have the ability to just even go to a public school and get a pretty decent education. There's a lot of room for improvement in the American education system. I'm not saying there isn't, But the fact that it's even accessible or will bus you from 30 miles out of the city to go to the school is unheard of. A lot of places you just wouldn't go to school. So
I recently gave a talk online for the Symbio for All Africa program. And then I caught up with, and so the talk for that is recorded on YouTube, so it should be searchable. Yeah. And I'm going deep into the science there with a scientific audience, for primarily scientists, biologists in Africa who were eager to learn more about synthetic biology. And then I had a follow-up call with a team of undergraduate synthetic biology students in Uganda who were particularly keen to get some advice on the project they're working on. And the constraints they're working within is a huge challenge. And for me, it's very important that people in constrained situations where they lack some access to consistent electricity in some cases, or consistent internet connectivity, or access to the best educational resources. Now we live in a globalized, connected world. If we're going to use social media for anything, instead of destabilizing democracies, we should be using it to expand out knowledge to people across the world. And so I'm trying to use social media and my platforms, online video communications, to benefit in that regard. But they're also lacking access to funds like research reagents. It takes a long time for research reagents they order from a company to arrive, and that's if they have the funds to buy it in the first place, because the universities have constrained funds. And this is not to even touch upon the issues, I'm sure, that exist of unequal distribution of access within the country, because I know from my experience in the UK and different places, like, access to those educational opportunities are not completely uniform. It's great that the US has different systems to help people, but it's not uniform, and different people have different abilities to interact. And then when you look at it at the global scale, like, you've got some places where, there's even more challenges. And so I do want to envisage a world and move towards that world where the barriers to education and these technologies are both lowered so that more people are able to access it, which I think will benefit humanity greatly because of the diversity of ideas, the more people working on difficult problems, which will then allow for more people entering that discourse of science that will hopefully lift everybody's work up.
I love it. I can't think of a better way to end, but I always end with, is there something that you want to share with the audience that you're either reading, watching, consuming? It could be related to this. It could be totally unrelated to this. We've talked all over the place, which is, I think, pretty typical of the podcast. I always like to acknowledge that.
Given the direction the conversation took today and talking about really weird things that might get me into trouble with some people over tentacles growing out of people, and I want to make it clear, you were the one who suggested that. I just said it's not breaking the laws of physics, so maybe, but ethically never. Ethically never. I just want to make this clear before I get cancelled by my fellow biologists for being unethical.
Oh, I'm sending it. We're live streaming to him right now.
Excellent. So, on that topic, it's been a little while, so I should go back and reread them, but the Oryx and Crake books by Margaret Atwood. So people might know Margaret Atwood because she's the author of The Handmaid's Tale, which is a phenomenal book, and a TV show has been made out of it. But for me, and The Handmaid's Tale, a fantastic book, fantastic examination of sort of like a dystopian, authoritarian regime. But her books are I think it's the Mad Adam trilogy, but it starts off with Oryx and Crake. And it's an examination of what synthetic biology, engineering of biology could do. And it is a very dark view. It has a very pessimistic view. And I think a lot of that comes down to how society and governments choose to regulate that. So it's optimistic in terms of the technology that you could give somebody a tentacle arm. It's pessimistic in terms of if you started doing that without proper ethics, with capitalism run unchecked, then what is the negative consequences of that sort of world? And so it's a very extreme view, but I do think it's a fascinating story. It's fascinating sci-fi, and it brings in some of the things we've been talking about, but a very dark view of it. And I want to see the optimistic view of it. I want to write the optimistic fiction of this. There's a genre of art called solar punk, where you get to see beautiful buildings that integrate with organic matter trees. You get to see green energy, wind turbines, all these wonderful things happening. And that's the future I want to lean towards. But I think it's important for us to read about the sort of dystopian visions that this technology could take us down to, and be well read on the topic. And it's also just a fun story as well.
That's awesome. Yeah, I agree. There is no such thing as like a good or bad technology. It's always how it's applied or how people use it. And I know some people get upset with that or they are uncomfortable with it, but that's the reality. Is their morality, ethics, largely a human invention, ethics a little hard. You could argue differently on that. But if you're not making people aware of the dark side, it's going to happen. And if it's toxic positivity is something I've talked about on the podcast before too. If you're only looking at the good side, you're not going to see the maybe unintended consequences. And I think AI is a really great example of this. AI is very magical for what it is. Generative AI is fantastic. It also takes a ton of power, right? And we're starting in that pollutes in that. And there's, for every positive, there's a negative. It's pretty much, I think, a universal truth. But we have to have those conversations. We have to be comfortable being uncomfortable. And that's also why I love, and I really appreciate you go into the tentacle arm thing, because a lot of biologists would be like, that's crazy, right? I'm not even going to talk about it. And I hope folks who have listened to it understand that partly it was a joke and kind of a crazy thing for me to suggest, but that doesn't necessarily mean it's all crazy either. As I die, coughing in the background. And it's having those conversations that hopefully get people thinking about it and thinking about that's bad. We shouldn't do that. Exactly. Right? We do that. That's the ethical thing to do, is to not try that.
And I think if humans are so evolved and so complex, then we should be able to use our power of ethics to not just invent something, but also be mature enough not to use it.
Right.
And some technologies are easier to do harm with than others. And when we obviously the atomic weaponry, nuclear war, this is something that is very scary and something that still gets talked about today. Like, we're still in a world where we could destroy ourselves through nuclear weapons. Instead, we're slowly destroying ourselves with climate change. But these are decisions humanity is making, and there's plenty of voices within humanity are saying we should never use nuclear weapons under no circumstances. We should stop climate change. We should stop pollution right now. And there's different forces at play that are driving these forwards. I hope that humanity, as a species, as a society, as a global society, will prove themselves mature enough that we are able to self-regulate. I just hope that's true, because at the moment things are looking pretty bleak for humanity's ability to self-regulate and share this world together in a mature way. And I just really hope that we turn out to be more mature than we're currently acting long term.
All right, we'll leave it there. Thank you so much. I appreciate you sitting on here this long and also sharing everything. Excited to have you back on after you've got the lab up and running a little bit more and make some other cool space plant technology for us to hopefully solve some really cool problems.
Yeah, looking forward to it. It's been a pleasure.
You can find us at 3reate.com. It's like the word create, but with a 3 instead of the C. As always, we encourage you to listen, watch, do. We'll see you next time. You can support this podcast at patreon.com/3reate or at ko-fi.com/3reate. Thanks for listening.